It is my great pleasure to introduce Dr. Karen Arndt today. Karen is a full professor at the University of Pittsburgh. She did her PhD in Michael Chamberlain's lab at Berkeley, and then did her postdoc at Fred Winston's lab at Harvard. And over the past past like 20 years or so, Karen's lab has been making tremendous findings on transcription elongation and chromatin, and how the PAF1 complex, and for example, is regulating these processes. She's also a member or a fellow for the American Academy of Microbiology, and she is also a AAAS fellow. Karen is also one of those people who is able to do everything. She's able to mentor grad students. Has been a director of graduate studies. She's just able to help everyone, but also still do amazing science. Those of you probably already know, I actually did my PhD in Karen's lab, so I can attest to her amazingness in mentoring. And now every time I tell people that I came from Karen's lab, they are immediately impressed. So without going on too much, I will go ahead and let Karen take it away.
Thank you, Christine. Thank you for making me blush here in my office all by myself. But it's a pleasure to be here today, and I really want to thank the organizers for this really amazing seminar series that you've put together. Hold on one sec. I need to get out of this mode so I can see my slides. Okay. Okay, so today I'm going to present some published work on our role of understanding of the PAF1 complex and its role in histone modifications, and also some unpublished work on how we've identified a new recruitment strategy for the PAF1 complex. So this is an introductory slides that all of you are aware of, but it reminds me to tell you that transcription takes place in this very complicated chromatin environment, and cells are armed with a diversity of strategies to overcome the obstacles posed by nucleosomes. For the talk today, we'll be focusing more on histone modifications, and you're well aware that all of the histones can be heavily post-translationally modified, and these various small chemical groups, or in fact a protein in the case of ubiquitin, can greatly change the structure of chromatin. So we are particularly interested in a histone modification on histone H2B towards the C-terminal end, and this is the monoubiquitination of a lysine 123 in Saccharomyces cerevisiae, which is our model system, or 120 in humans. This monoubiquitin does not lead to destabilization of the histone, but instead serves as sort of a signal to change the nucleosome and allow the recruitment and the activity of histone methyltransferases that modify lysine 4 or lysine 79 on histone H3. And I'm sure you're well aware from this series, from Cynthia Wahlberger gave a beautiful talk earlier this summer describing how this modification on H2B leads to the positioning and the activation of the enzymes, the methyltransferases that target H3. But we're really interested in how this mark is deposited, and that's because we are interested in understanding the functions of a transcription elongation complex called the PAF1 complex, and I'll introduce you to that a bit more in the next slide. But I just want to point out that the PAF1 complex is just one of many transcription elongation factors that help change the structure of chromatin as RNA polymerase traffics along a gene. And some of these factors are involved in depositing histone modifications in various patterns. So for H2B ubiquitination, what's been observed is basically a more or less constant level across the gene, dependent on the level of gene activity. So more highly expressed genes, you see higher levels of this modification. Work from many labs, including Steve Bertowski's lab, have demonstrated mechanisms by which some of these methylation marks are restricted in more domains across the genes. And of course, there's also modifications that are not H2B ubiquitination dependent that are also observed, such as K36 trimethylation. So some of the work that we're doing in our lab is focused on understanding how these modification patterns are determined, and particularly focusing on H2B ubiquitination. What are the factors involved? How do they work together, and how do they target chromatin? The PAF1 complex is a transcription elongation complex first identified by Judith Janing's lab in a Pol II affinity column, and thus its name, polymerase associated factor complex. It's a 5 subunit complex shown here. It's conserved from yeast to humans. We started working on this complex way back in the day when I was a postdoc with Fred Winston. I identified this gene RTF1 in a genetic suppressor screen for TBP mutants, and our follow-up work demonstrated it was part of this elongation complex. So what does this complex do? Well, first off, it travels with Pol II on all active genes in the genome. So it's one of the globally acting transcription elongation complexes. And what it does there, in part, is help establish various histone modifications, in particular H2B ubiquitination and its downstream methyl marks. It is also important for the recruitment of a chromatin remodeling factor called CHD1, which plays a role in spacing nucleosomes. And we discovered this a few years back in a 2-hybrid search for RTF1 interactors. And these combine, of course, together to change the structure of chromatin on genes. A few years earlier than that, we discovered, along with Judith Janing's lab, a role for this complex in transcription termination. So mutations in certain subunits of this complex lead to the read-through of terminators, specifically those for non-coding RNAs that use the Saccharomyces cerevisiae NERD1 NAB3 SEN1 complex. The Janing lab and also the Dinlac lab demonstrated a role for this complex in proper poly-A-dependent termination as well. And then back to metazoans, a number of groups, including the Kramer, Rader, and Schlotterfer's labs, have investigated the role of the PAF complex in establishing and releasing the promoter proximal pause polymerase. And then finally, Jane Meller's group has found a post-transcriptional role for this complex in mRNA export. So this is a complicated transcription elongation complex. And what we've been trying to investigate through both genetics, biochemistry, genomics, and structural biology are what are the various functions, what are the direct functions. It's difficult to tease apart from null alleles how many of these functions are actually direct or indirect. So today I want to share with you a story that we published a few years ago on how the RTF1 subunit is critical for the deposition of H2B ubiquitination. And then toward the end of my talk, I'll talk about the tethering points that keep the PAF complex on the polymerase. So how does this complex play a role in H2B ubiquitination? Well, that was the mystery we set out to address, partly because it's not an enzyme. There's no known enzymatic functions to it, yet we already knew the enzymes, and this is the ubiquitin conjugase and ligase, Rad6 and BRE1. So these are well known to deposit ubiquitin specifically on H2B, yet in cells lacking the PAF complex, you see very little, if any, of that modification. This modification is not only a prerequisite for downstream modifications, but in its own right regulates transcription. It also has implications for DNA repair, chromosome segregation, and therefore genome stability. And there are many implications for cancer development. So one hypothesis is that this is just a recruiter who rides on the polymerase and drags in these other factors, but is that all that it does? So we started this story, again, a few years ago by basically using our genetic tools. And so we deleted each of the subunits one by one in the PAF complex, and it's very clear by this Western blot that only the RTF1 subunit is essential in vivo for H2B ubiquitination. Now, these subunits, PAF1 and CTR9, are clearly also important for that. And we don't know exactly to what extent they're directly involved or whether it's because when you delete them, you actually destabilize the complex in its entirety. They're structural cores of the complex, as shown by beautiful cryostructures that came out from the Kramer lab recently. So here, RTF1 seems to be the business end. And for we've subjected this particular subunit to a great deal of mutagenesis, both through unbiased genetic screens and also more directed approaches. And what we can say is that this one subunit in the PAF complex does a lot of things. So it's tethered to the rest of the complex. It has a large central domain that I'll mention again later in the talk that is involved in recruiting the whole complex onto RNA polymerase II. That is mediated through SBT5, which Inwa just introduced to you. There are two other domains of interest. There's an N-terminal region that's necessary for this binding to CHD1, which we're studying in a separate project in the lab. And then there's this histone modification domain that we identified. And what this is But this does is in some way make it possible for the path complex to stimulate all of these histone modifications, in particular the upstream H2B ubiquitination mark. So we know that point mutations or deletions in this region eliminate this. So now a number of years ago, a former graduate student in the lab, Marcy Warner, and then a technician, Tony Piro, set out to ask whether this domain was not only necessary but sufficient. And so they performed an experiment that I thought was really crazy, but basically what they did was they expressed just 89 amino acids of a 558 amino acid protein in yeast, or even smaller, 66 amino acids from the center of this domain. And they expressed that on a plasmid hooked up to an NLS and an epitope tag. And they took out the full RTF1 gene from the genome. And what was remarkable to me was that when they did that, they could fully recover global levels of H2B ubiquitination, either with 66 or 89 amino acids or full length RTF1. So the fact that you could replace the entirety of this subunit with 66 amino acids struck me as a bit unusual if we were to consider only a recruitment platform idea for the PAF complex. We further showed that if we delete the entirety of the complex through a quintuple deletion strain, 66 amino acids is all you need to bring back H2B ubiquitination to the cell. So the stimulatory role of the PAF complex can really be boiled down to 66 amino acids of this very large complex. So how is it working? And so we did a lot of different biochemical and genetic experiments, but I just want to bring out one because this is a technology that we like to use, and I think it's of general utility, and it's worked for us pretty well in at least 3 different circumstances. So what we've done is we've tried to identify the proteins that interact directly with this domain. And so what we used was a strategy developed by Peter Schultz's lab, who has done this amazing job of expanding the genetic code in various species, including yeast. And so the way you can expand the genetic code is to make use of amber codons. And you can make use of amber codon suppression with specialized tRNAs that become charged with specialized amino acids. So looking over here, we deleted the RTF1 gene in the genome, provided an RTF1 derivative on a plasmid, which will have an amber codon mutation at a particular point in the HMD domain. And we transformed that strain with a plasmid from Pete Schultz that carries a specialized tRNA for recognizing amber codons and is chargeable by a specialized aminoacyl tRNA synthetase that will target this novel form of phenylalanine onto the tRNA. This novel form of phenylalanine is called BPA. It's photoreactive. It will go into the protein in vivo by translational read-through of the stop codon. And now you have that in your protein inside cells. And because it's photoreactive, you can shine a UV light right on top of your cells, right on your bench, and you can induce direct protein-protein crosslinks. So that's how we set about identifying what this domain is interacting with inside cells. And so this is just a sort of a proof of principle here. We introduced BPA throughout this domain. We shined a light on the cells, and here's an example. When we grow the cells in BPA, we now get full-length RTF1 protein because we have translational read-through, and when we put a light on the cells, we can cross-link something. So in this Western blot, we're probing from the bait. So what is the prey? That is always the problem with these experiments. But for this particular histone modification, we had some candidates. And I can show you here that one of the things cross-linking directly to this domain, either within full-length RTF1 or when we just express the little domain on its own, what we cross-link is the E2 RAD6, which is actually the enzyme that puts ubiquitin onto the nucleosome. So this tells us that this transcription elongation factor is very directly involved in interfacing with the enzymes that put ubiquitin onto the nucleosome. We're fortunate to have Andy Vandemark next door. He's a crystallographer and who trained with Chris Hill and Cynthia Wahlberger. And so Andy solved the crystal structure of our little domain from RTF1. The yellow residues are those with which those we identified through unbiased genetic screens that are important for function. When you map onto this where RAD6 is cross-linking, which we can identify by where we put BPA, we can see that it's cross-linking on top of this large helix. And in a project I don't have time to tell you about today because we're still working on it in the midst of this pandemic, is to identify what part of RAD6 is actually in touch with this domain. And hopefully I can tell you about that later. So one thing about identifying this direct physical contact and this small domain as being pivotal led us to suggest that maybe this is actually more of a cofactor, that the elongation factor is providing a domain as a cofactor in some way engaged in the chemistry. And so we decided to reconstitute the H2B ubiquitination reaction in vitro with recombinant factors. And we're helped a lot here with some of our colleagues in the chromatin field. So we obtained recombinant nucleosomes from Songtan and recombinant yeast BRE1 and E1 made in baculovirus from Jae-Hoon Kim's lab. And we prepared Rad6, ubiquitin, and then the purified domain from RTF1. And what I want to point out here is if you leave that HMD domain out of the reaction, so just the basal reaction here, in vitro you detect H2B ubiquitination. And this is something that you won't see in vivo because as far as we can tell, in vivo, the reaction is highly dependent on the PAF complex being present. I think detecting it in vitro might relate to the fact that we can really fill up the Eppendorf tube with high concentrations of these guys. When we add in the HMD, however, we can see a 4 to 5-fold stimulation of the reaction. So that suggests that this is actually directly working into the nucleosome mediated catalysis in some way, whether it's affecting catalysis or affecting binding or positioning is something we're really actively studying right now. And I do want to point out a control over here. This is a point mutant in this domain that we identified years ago in an unbiased genetic screen. It's dead for ubiquitination in vivo and it cannot stimulate in vitro. So these data together suggested to us that RTF1 as part of the PAF complex provides a domain which acts some way as a cofactor actually in making sure all of the reaction takes place properly on the nucleosome. I didn't tell you this because I don't have time, but we did a lot of chromatin IPs and chip exo experiments together with Frank Pugh and ask where does the ubiquitination go when we just express our HMD domain out on its own in the cell. And basically it goes where it shouldn't go. It goes to untranscribed regions of the genome as well as transcribed. And so we believe that the reason you get ubiquitination of H2B on the genome is because you're tethering, one of the reasons is because we're tethering this important domain to the polymerase. And so therefore these connections to the polymerase are really important in governing where this mark goes. So getting back to the nucleosome, though, we were curious as to know what its role would be as a target for some of these factors, in particular Rad6, BRE1, and RTF1. And so here I get a chance to tell you a little bit about Christine's work when she was finishing up in the lab. So what's the role of the nucleosome in this whole process? And so one thing that we did was do an unbiased genetic screen of all four. Actually, we used H2A and H2B mutant libraries on plasmids provided by Ali Shalataford, and we screened all of the H2A and H2B viable mutants in a genetic screen that I don't have time to tell you about, but it involved a phenotype that we can score for those that might be defective in H2B ubiquitination. And what Christine and an undergrad, Ali Young, found was that there are certain positions on the nucleosome surface that when mutant, you lose the ability to support proper H2B ubiquitination. So this was our attempt at identifying how these factors will engage with the nucleosome. And when you look at the nucleosome, you're all well aware, I'm sure that there's this really wonderful acidic patch that is available for recognition by a huge number of different factors now. So Christine's mutants all map to this, basically all map to the acidic patch. And so this said to us that this surface is somehow serving as a recruitment or docking site for some of these proteins involved. And in fact, Alwyn Kohler's lab had around the same time demonstrated that he could get some cross-linking between the E3, BRE1, and this region of the nucleosome. So Christine was a very brave student, still is, I'm sure. And what she decided to do, because she's ferocious at the bench, was decide to actually incorporate this cross-linking competent BPA residue on the surface of the nucleosome and put that into living cells. And so if you can do that, then you can imagine shining a UV light on those cells and then catching what might be binding to your favorite part of the nucleosome, and in this case, the acidic patch. So Christine worked really hard to identify two positions in H2A, A61, Y58, that when you change those to BPA, we could produce full-length H2A in the cells, and we could also see some cross-linking. So I did want to point out one thing about this particular experiment. While we normally like to do with these BPA substitution experiments is have that as the only protein in the cell. But for nucleosomes and histones, we were concerned that if we didn't get enough amber codon suppression, we wouldn't get enough H2A and the cells would basically be dead. So Christine produced an ectopic copy of H2A that was BPA substitutable. Okay, so here probing from the H2A side where she has an epitope tag, and these are the most beautiful gels still I think we've ever run. And you can see cross-linking of H2A from to various proteins. And I always get asked about what these proteins are, and I really can't tell you that I know exactly what those proteins are because what we really need to do is scale this to a more mass specable level. Um, Most of the time right now we've been doing Western blotting, but we have tried some mass spec and Christine was pivotal in that. But I do want to say that for A61, we do know what one of these bands is, and that's H2B. So if you probe with H2B by Western, you can see a cross-linked species. And if you look at the nucleosome structure, you'll see A61 is right next to H2B. So that was a nice proof of principle, but we were interested in H2B ubiquitination, and we were amazed the day that Christine found that, in fact, what is directly cross-linking the acidic patch is RTF1, so the subunit of the PAF complex that carries the histone modification domain that we talked about is now directly cross-linking to H2A in the acidic patch. And this fit really nicely with previous work Christine had done where we can take viable H2A substitution mutants in the acidic patch. They're not all viable, but these guys are. And as the only form of H2A in the cell, we can ask, well, what happens to the occupancy of RTF1 on the genome? And so this CHIP assay shows you that when we make changes in the acidic patch, we reduce RTF1 occupancy. So you might be asking, well, okay, you know RTF1 is cross-linking, but what about that little domain? So it's a pretty hard experiment to pull off, but Peggy Shire in the lab is amazing at generating novel yeast strains. And so what Peggy did is she replaced the full RTF1 in the yeast genome with just that little domain, the HMD, under the, in this case, for good measure, we put it under the control of an inducible promoter. And so what you can see down here in this Western blot that is that in the presence of the BPA substituted histone, in the presence of galactose, we can see cross, and in the presence of UV, under only in those conditions do we see cross-linking. So this tells us that the HMD domain is that part of RTF1 that is in direct physical contact with the nucleosome. And we think, therefore, that what RTF1 does and the PAF complexes play a very important role in putting together, I like this jigsaw puzzle idea, where this all has to somehow fit together, right? So we already knew that RAD6 and BRE1 interacted as E2s and E3s do. We found RAD6 and RTF1 directly physically contact one another, and RTF1 also directly physically contacts the nucleosome. And the Kohler lab found this contact. So somehow this all comes together in what is an efficient way to ubiquitinate, and specifically ubiquitinate H2B. So one thing I didn't get to point out in much detail is over here that through work we did with Frank Pugh's lab that, by constraining this HMD onto the polymerase, I think that is really important in determining where this modification goes. Okay, so I now just want to, for the last part, talk about some unpublished work where we're looking at how the PAF complex is coupled to the polymerase during elongation. Because if we're going to constrain that domain, we have to constrain it through some other connections. That domain is not a Pol2 interacting domain. In fact, we knew from our previous work that there is a large section of RTF1, it's known as the +3 domain, that is. So we demonstrated through work with Andy Vandemark that domain directly contacts SPT5. And SPT5 is a universal transcription elongation factor. It's essential for viability. And what's interesting about SPT5, among many aspects, is it has a C-terminal repeated domain, not so unlike the C-terminal repeat domain of Pol2. It's phosphorylated, and more and more work is coming out on that. But what we found is, in part through crystallography with Andy Vandermark, is that the +3 domain of RTF1 directly contacts the C-terminal repeat of SBT5, and it does so through a phosphospecific manner. And we made mutations in this interface and showed that we weakened the recruitment of the PAF complex. So we know that this is an important interaction. But looking back at the literature, it was clear that that wasn't the whole story. So Judith Janing years ago deleted this subunit, CDC73, from yeast and found less occupancy of the PAF complex on the genome. And Alan Hinnebusch's lab, doing some biochemical experiments, found that CDC73 can bind to CTD, Pol2 CTD peptides that are phosphorylated. And so that led us to wonder a bit about CDC73. And we're really interested in that protein for a variety of reasons. It's very important actually in human health. It's a known tumor suppressor in humans. So one thing we did early on was look at the domain structure of CDC73. And so it has a very highly conserved C-terminal end that we deleted in yeast from the genome. And then we asked, well, what happens then? Do we lose PAF complex on the genome? What happens to the complex? So looking at the complex, we tap-tagged both the full length and the C-domain deleted form. Both of those versions, using a tap purification, both of them interact with all of the rest of the subunits of the PAF complex. So we don't think that this domain is really holding the complex together. In fact, we think it's the N-terminus based on published literature. But if you do chromatin IP experiments where you delete off this C-terminal domain, this highly conserved domain, you see less PAF complex on the genome as shown here by chromatin IP. The polymerase seems to be not greatly affected by doing that. So again, we're fortunate. Our next door neighbor, Andy Vandermark, is awesome at crystallography, and he solved the structure of this domain. This domain actually forms a RAS-like fold, kind of like the RAS oncoprotein, but does not have the necessary binding pockets for nucleotides. So it's just a fold that's very RAS-like. But what's nice about a crystal structure is it can help guide your future experiments. And this, as I mentioned, CDC73 is known as parafibromin in humans, and it's important for the prevention of various heritable cancers. Okay, so here we have this domain, and I always have a lot of undergrads in the lab. And so one of my undergrads, Ellie Kerr, decided to incorporate BPA, again, the cross-linking competent phenylalanine analog, onto the surface of CDC73, express that in yeast, expose the cells to UV light, and see what happens. So the crystal structure helped us in that regard because we could focus on sort of surface exposed residues, some conserved, some less so. And so what Ellie found, for example, 300 out here, is that you can produce full-length CDC73 in the presence of BPA, and you can cross-link something to it. Again, we're probing here from the bait. So what's in here? So here we didn't really have a clue. However, Ellie did this amazing thing one day where she just ran the gel forever, it seemed. And what we found was this 300 BPA substitutions, normally an arginine in CDC73, gave us two very clean, precise cross-linked moieties here. And Alan Hinnebusch's work had suggested to us that the big one here might be Pol2, and in fact, that is a direct physical contact inside cells of CDC73 and RPB1. I'm not showing you here what, but we actually used a derivative of RPB1 with a TEV protease cleavage site to lop off the CTD after cross-linking, and the cross-link goes with the CTD and not the core domain of RPB1. But what about this other band? Well, it's a big protein. There are big elongation factors in yeast, and one of them is SPT6. And in fact, that's what that is. So when we probe from the SPT6 direction, it is the smaller of these two cross-linked species that is reactive with the SPT6 antibody. So this is a direct physical cross-link between CDC73 and SPT6. And we can test the relevance of both of those cross-links, so Pol2 and SPT6 cross-links, by incorporating another mutation into CDC73, in this case, a substitution at this tryptophan that we know confers transcription defects in vivo. So these transcription defects include things like SPT phenotypes. And so when we make that mutation, we lose our cross-links. So we believe this is physiologically relevant. So what is SBT6? I'm sure you've heard about it before. SBT6 is one of several essential histone chaperones inside yeast. It's a highly conserved protein. It's got a very complicated domain structure. It's quite large. It's over 1,400 amino acids. And Chris Hill's lab and Patrick Kramer's lab have given us really beautiful structural information. It has the only known tandem SH2 domains in yeast, and these actually directly contact Pol2. So this is a histone chaperone, which means it's in this case it's bound to the polymerase and it's involved in making sure histones and nucleosomes are redeposited properly following movement of polymerase through the template. Without that, you get cryptic initiation on the gene bodies of genes within the gene bodies. So the question for us is, well, how important is this crosslink we identified? You know, is there any role for SPT6 in governing the recruitment of path onto the genome? So my graduate student, Mitch Ellison, incorporated an auxin-inducible degron on the C-terminal end of this essential protein. After exposure to auxin, we can greatly deplete SPT6 levels. In about 60 minutes, it's completely gone. In that time frame, we do not reduce the viability of the cells, and you can see we don't change the levels of PAF1 either. As expected, with ChIP-seq experiments upon treatment of cells with auxin versus our DMSO control, we see great depletion of SPT6 on the genome. What you're looking at here in these profiles are genes partitioned according to transcriptional activity as gauged by RBB3 occupancy. So the very dark line are the most highly occupied genes with respect to the polymerase, so the most active genes. So great depletion of SPT6, that's not surprising, we're depleting SPT6. What about PAF1? We see a dramatic loss of PAF1, again, on the most highly expressed genes. And so this argues that, in fact, SPT6 does play an important role in the occupancy of PAF1. RNA polymerase and SPT5, on the other hand, are not as severely impacted when we reduce SPT6 levels in the gene through depletion. And in fact, they're very similar to one another. These guys correlate in a correlation analysis, and these correlate in a correlation analysis. So this strongly indicates SPT6 is recruiting or playing some role in maintaining occupancy of the PAF complex on the genome. But is this a direct interaction? And so what Mitch did was a lot of biochemistry, and he made recombinant SPT6 with an M Ruby tag on it, and recombinant CDC73 with an M Clover tag. And he used those in native gel shift assays as shown here. So every lane has the same amount of M Clover CDC73. And if you look down here, if you add SPT6 to just M Clover alone, no CDC73 attached to it, you can separate SPT6 is Ruby tagged from M Clover, so that's a control. But if you now attach CDC73 to M Clover and you can titrate in SPT6, you can see them starting to interact as indicated by the shifted species. And this can be quantified, and we can do that both with wild type CDC73 here in black and a mutant form. Again, the tryptophan 321A mutant has less affinity. And so this is in the order of nanomolar scale. Mitch did a complementary experiment and used fluorescence anisotropy. And using fluorescence anisotropy and his recombinant fluorescently tagged proteins, again, we can see a direct physical interaction between the C-terminal end of CDC73 and SPT6. He then moved on to chemical cross-linking in vitro with the help of Yang Shi's lab at our School of Medicine. And this allowed him to find quite a few chemical cross-links, most of them coming from the C-terminal end of CDC73. And then they're reaching to various domains within SPT6. Which makes no sense if you look at it like this, but if you actually model this onto the known structures of SPT6, what Mitch has been able to do using modeling platforms is come up with a hypothesis on how the C-terminal end of CDC73 could be docking onto SPT6 through this extensive interface here. And this interface is now something we're probing using mutational analysis. So to end this part, what we have then are at least two, probably three mechanisms that help maintain the PAF complex on the genome, and how those different mechanisms interface with one another. For example, if they're gene specific or position specific is something we're now set up to test with all of our mutants. One of those attachment points is through SPT5 and RTF1. The other attachment site is between CDC73 and SPT6. And also, I do want to point out, we did find the direct physical contact with the CTD of Pol2 as well. So to end this, we've identified what we believe is a new mechanism for retention or recruitment of the PAF complex onto genes during transcription elongation. And again, we're probing this now guided by that structural information. And I do want to point out that this is a part of CDC73 that was not present or not visualized in the recent cryostructures from the Kramer lab. So we're quite excited about the fact that our unbiased cross-linking has led us in this direction. I need to thank the people in the lab who've done this work. So this is us at Penn State last summer at the Chromatin meeting, and Christine was already in Toshi's lab, but she was at the conference as well. This is Mitch Ellison, who did all of the work on SPT6. This is Peggy Shira, who's really great at all the BPA cross-linking in the lab. And Brendan McShane is a recently departed technician who's been working on the RAD631 project. He also helped Christine with some of the histone cross-linking experiments. I have a lot of undergrads to thank. Matt Blacksmith has helped along the way, Ellie and Rachel and Allie. I have some wonderful collaborators as listed here. I do want to highlight my gene expression group in my Department of Biological Sciences, Craig Kaplan, Myler Lee, and Sarah Hayner, who I think just spoke here recently. These are our funding sources. And this is us having milkshakes together. So I'm happy to take questions.
Thanks, Karen. That was a fantastic seminar, and my cat joined us too. Um, I'll go ahead and start pulling in the questions. And as we mentioned before, if you want to raise your hand to ask a question, feel free to do that. The first question is from Apurva again. So he asks, since the effects of SPT6 depletion are dependent on transcription levels, does depletion of SPT6 affect A, H3K36 trimethyl levels across gene bodies, B, cryptic initiation within genes, and C processivity, and D, antisense / read-through of Pol2. And we can do those stepwise again if you.
Okay. So actually, a lot of that has been addressed in the field. So with respect to K36 methylation, Brian Strahl's lab has demonstrated a role for SPT6 in that, and so has Fred Winston's lab. With respect to, I think you meant, so like things like cryptic transcription across the gene body if you deplete SPT6. So we haven't yet looked at transcription in our cells. Fred Winston's lab had a really nice paper in Molecular Cell, I think in 2018, looking at SPT61004, which is a really important SPT6 allele. Many people have looked at it. And that mutant does show lots of evidence of cryptic initiation within gene bodies. What else was there? Termination. We haven't looked at termination and poly-A termination. Did I miss something?
I think that's it, and then also just processivity.
Oh yeah, I don't think SPT6 has been considered to be a processivity factor in the way that SPT5 has. No, I'm not definite about that, but I don't think so.
He also has a follow-up question, which I think is interesting. He asks, in humans and some cell models, SPT6 and SPT5 are recruited to Pol2 by MYC. However, what recruits them onto Pol2 in yeast where MYC is not present? He's very curious.
Oh yes, we're all curious about these things. No, I think that's a great question. Well, with SPT6, I think there's really convincing evidence from Chris Hill and others that there is, the Bertowski labs contributed to this as well, So what Chris Hill has discovered is there are residues in RPB1 in the linker domain between the core, so the body of RPB1 and the tail, that are phosphorylated by BUR1, which is the CDK9 analog in yeast. Phosphorylation of those linker those linker residues in RPB1 leads to the recruitment of SBT6. So there's beautiful crystallography showing the binding of SBT6, tandem SH2 domains to the linker of RPB1. And recently, Burtowski's lab showed that that was phosphorylation dependent by BUR1 CDK9. That's it for SBT6. I don't know if I want to get into the SBT5 story. I'm not sure I know that one quite as well. But at least there's a phosphose switch.
Right.
But whether there's activators like MYC involved in yeast, I think is an open subject.
Yeah, it'd be really interesting if someone
Exactly.
found that. We do have another question before we wrap up for the coffee chat. Oh, there's even more, but I will take only 2 more because we are running out of time. So Akash Grover has asked, is it K123 acetylated, and in any of in vivo situation, could this affect RTF1 function?
Oh, I think it might be
It
acetylated.
is, right? I feel like
I think it
we
is
yeah.
acetylated. I don't think we know much about that.
Yeah, I feel like there's an antibody in it for it, but I don't remember if
Yeah,
it's working.
that's a good question.
Mhm. And then Zulong Chen asks, how is the NLS HMD fragment specifically rotate or recruited to H2B for ubiquitination, and can it interact with Pol2 independent of the other path components?
So it can't, and as far as I know, it can't interact with Pol2, but it can, what I didn't show you is it can interact with nucleosomes directly. So we can do EMSA assays with pure nucleosomes and that little HMD, and it will bind to it. So I think it has some affinity for the nucleosome. I don't know how. That's something we would like to probe structurally.
Cool. Thanks, Karen. Thanks, Inwa, for wonderful talks.